First of all, thank you very much for the invitation. I've been really enjoying the conference, and it's great to start talking about the human microbiome in an Exposome conference. So the human microbiome, as most of you may know, is a collection of microorganisms that reside in and on our bodies. We have roughly as many bacterial cells as human ones in our bodies, and where they reach the highest densities is in the gut. So this is one of the reasons, together with their wide range of functions, why we mostly focus on the gut microbiome. Also because it's been associated to a wide range of conditions, including those in neuropsychiatry and neurodegeneration. So should we talk about the microbiome in an Exposome conference? Well, in the 2005 definition, the microbiome was not mentioned. Now I'm taking here a figure from the keynote of the first day, where that is mentioned. But what I would like to claim is that, yes, we should look at the microbiome as something that is related to some of our health outcomes. But on top of that, the microbiome is also modulated by many other components of the exposome, not only diet, as maybe the most obvious ones, but also psychosocial and behavioral factors. I'm going to elaborate on that later. And in this way, the microbiome can also be a mediator between many of the factors in the exposome and our health outcomes. So the gut microbiome varies throughout our life. We get our first inhabitants of the microbiome during delivery and short after. Then during adulthood is roughly stable, and in the elderly, we start losing diversity. And there are different factors that have been identified that modulate composition of the microbiome in these different stages. So when I started to work on this, which was a bit more than 10 years ago, there were many studies showing microbiome differences between case and controls on many different conditions. And one doesn't have to be very smart to say, well, yes, the disease could be explaining these differences, but also many other factors like diet or medication. It's something that in the exposome field that's obvious, but in these first publications on the microbiome, it was not always assessed. And also they were often very small. So yeah, power was also an issue. So I was working in Belgium at that time, and we set up the Flemish Gut Flora project, where we tried to get the population level overview of what was the composition of the gut microbiome. At that point, with a bit more than 1,000 samples, you were allowed to say you are doing this at the population level. And that's what we had, one of these first publications that look at the composition of the microbiome in the population. So as a first step, we tried to look at what's the exposome of the microbiome. So which are the factors that are associated with microbiome composition? And we found that, first of all, had some of the obvious suspects like age, sex, body mass index, or some blood markers that were associated with inter-individual variation in microbiome composition. But there were also some ones linked to mental health, like clonazepam, benzodiazepine, or we also had in there chronic fatigue syndrome or irritable bowel syndrome. I must also say that overall, with all our questionnaires, markers, we got to less than 8% of variation explained. So there is still a lot that we don't know. Sorry, this is changing on its own. Yes, we also achieved a 90% replication rate on the lifelines, which was a similar cohort from the Netherlands. And so we said, okay, let's look at the association between the microbiome, mental health at the population level. So what we had in the Flemish Gut Flora project was the RAND, Health-Related Quality of Life questionnaire, which is mental health and physical health indicators. And then we also had diagnosis of depression for a bit more than 120 individuals, a bit less than half being on antidepressants. So our first step on this first study at the population level, on the association between microbiome composition and mental health was assessing what percentage of inter-individual variation in microbiome composition was associated with quality of life indicators and depression as compared to the main known covariates. So anthropometrics, where we had age, sex, and BMI, or gastrointestinal factors, where we had Bristol-Stool score as a proxy for gastrointestinal transit time, and then gastrointestinal disease. And what we saw in a cumulative model here is that the anthropometric and gastrointestinal factors enter the model first, but also quality of life related factors like body pain, physical functioning, and general health were entering this model. And then there was also a diagnosis of depression. So I explain a smaller portion, but still it's not fully redundant. Then we looked at it more in detail at the genus level, and we went for associations between microbial general and quality of life and depression, accounting for these main covariates. We found mostly that Faecalibacterium and Coprococcus, two bacterial genera that are known to produce butyrate, were positively associated with quality of life scores, whereas Flavonifractor, a bacterium that has been reported to grow on GABA, was negatively associated. Then on depression, we also found Coprococcus again and Dialister that was in reduced relative abundances and in patients with depression as compared to the rest. And these associations were also holding when taking into account the influence of medication, and they were also reported before in the literature based on smaller case control studies. And here, the bubbles with a tick, they were replicated in the Lifelines deep dataset again. So there were some more publications on the gut microbiota in depression and anxiety, and overall what we see is that there are lower relative abundances of short chain fatty acid producing bacteria together with an increase in tax associated with inflammation. And this would make sense mechanistically, as in a healthy state we would have these short chain fatty acid producers promoting homeostasis, where in the inflammatory state that characterizes depression and anxiety, we would have a higher abundance of inflammatory species, which compete with the short chain fatty acid producers. There is more bacterial translocation, and this is also part of this vicious cycle. But of course, this is just us hypothesizing on the mechanism. So when we go into more detail on looking at the pathways involved in microbiome gut-brain signaling, we can think of neural, endocrine, or immune routes, but probably what's the most direct communication and also one that we could potentially modulate is the microbial metabolism of neuroactive compounds. So members of the microbiome can produce and degrade short-chain fatty acids like butyrate, acetate, propionate, but also hormones, cortisol, immunomodulators, or many neurotransmitters like serotonin, dopamine, or GABA. All of them can be produced and degraded by members of the gut microbiota. So we see bacterial metabolites as potential functional mediators of the microbiota gut-brain axis. Through this metabolism, these compounds could then signal to the enteric nervous system or the vagus nerve, but they could also translocate, so crossing the gut-blood barrier, some even the gut-brain barrier, many not, or just have immune modulatory effects. So when we study the composition of the microbiome, traditionally we would have fecal samples from which we would do the extraction, and the traditional approach was 16S ribosomal RNA sequencing. So just sequencing some small areas of the 16S gene. This would give us information on the taxonomic composition of the microbiome, so mostly the general, and in some cases we would get species resolution. But you may know that, for example, just if one E. coli, there are some E. coli strains that are toxic, whereas others are perfectly commensal. So all of us have E. coli. If we just know we have the species, we just don't have enough resolution to know whether this would be linked to disease or not. So with shotgun sequencing, where field is moving now, or mostly we are there now, what we get is taxonomic composition on much better resolution up to the strain level. But on top of that, we also get information on the functional composition of the microbiome. We are doing this in an untargeted way, so we are just sequencing any gene that is out there. So we get information on actually the genes that are present. But that's far from easy to interpret. So what we did was developing a computational tool to target these microbial transformations of neuroactive metabolites and facilitate this interpretation of the microbial neuroactive potential from metagenomic data. This was a set of gut-brain modules defined just as the set of biochemical steps that follow the synthesis degradation of neuroactive compounds by the microbiota. So you have one example here for degradation of the neurotransmitter dopamine into the OPAC. So 2 steps, this is just defined here. And this led to a first catalog of the neuroactive potential of the gut microbiome. So basically, who does what? So that's something that is interesting because yeah, just this way, when we have one microbial strain, we can know what capacity it has of doing. We applied this to the Flemish Gut Flora project dataset, and one interesting association that we found is this positive association between DOPAC synthesis, so dopamine to DOPAC conversion I mentioned earlier, and mental quality of life scores. And also here, the association with social functioning was replicated in the Lifeline Sleep dataset. So this was published in 2019. The field is advancing fast, so there have been many more pathways that have been discovered. So now we are working on an updated version of the tool where we have now 190 modules for 70 neuroactive metabolites. We've expanded mostly on bile acids and hormones. As it was actually already mentioned yesterday, there is also growing evidence of sex hormones having neuroactivity. And there is this co-fluctuation between levels of sex hormones and the microbiomes. And that's, I mean, the interaction here could be direct, as many members of the gut microbiome can synthesize, degrade, conjugate, and deconjugate sex hormones. And that's true not only for the endogenous hormones, but also for the exogenous ones, so synthetic ones that we get through medication, or phytoestrogens, for example, that we get through diet. And this could have implications both for physiological conditions, but also for hormonal medication efficacy, and finally in pathological conditions. So we've expanded that recently. That's also a complex set of reactions. Some of them are shared with the human ones, but there are others that are actually microbial specific. And now we have this updated catalog of sex hormone metabolism by the microbiome. Just to say that the field is advancing fast, like one example is that in 2019 we had a bit more than 500 genomes, and we already thought that was a lot. Now we are at more than 1.6 million genomes for more than 100K species. So yes, we have many more features to analyze. That's complex, but at least we have a much better overview, as we still have like less than 1% of the microbiome that is cultivated in the lab. So we really need these metagenomic methods to get a better survey. So now we are applying these tools to survey the neuroactive potential of the human gut microbiome across neuropsychiatry, defined by the DSM-5 and ICD-11. So we are taking here any publicly available data set. So we thank those that are following fair practices. We are also involved in setting guidelines for getting standards where we can actually reuse those data. So many of these cohorts are relatively small, but we are then putting them together in meta-analysis, and we are getting finally some robust patterns. As when we go for individual publications, we see that actually what's shown in different cohorts is very different. We actually see that most of these differences are taxonomic, so in different populations there might be different species-level associations, but when we look at the functions of the microbiome, we are getting more robust signatures. So maybe what's really important is actually not really which specific species or strain you have in there, but what they have the potential of doing. So that's where we are trying to move forward to get to biomarkers. But besides biomarkers, there are also many, and this was captured from news and views that was published in our paper already on 2019, where they were saying, well, we might be in the early days where we can actually treat depression through targeting the microbiota. We are very far from that, I must say, still. But there are now, I mean, this year we had the first FMT, fecal microbiota transplantation studies on major depression disorder and bipolar disorder, where they showed that it was safe. We see that, well, at most, FMT would help as an adjunctive to pharmacological treatment, but as I'm saying, that's in the very early days. And when thinking about probiotics, so there are now many in the pharmacies, you can see the strains that are there are really not overlapping with those that we see missing in patients with depression. So that's really like not evidence-based. And if we want to think about evidence-based mechanisms, what we need is to understand still like basic questions, as it was said in other talks. For example, we still don't really know how we get our own microbiomes. And that's something that, well, I find it's key if we want to think about how to artificially induce getting new microbial strains. And we've been working on that a lot for the last years. The key methodological point in there is that if we look at the microbiome at the species level, we might just share around 30% of the species with anyone else. So that's not really giving us enough information. But if we look at this at the strain level, we share less than 0.1% of strains with people we have never met. So we are using strains as the unit to track transmission of the microbiome. That's something that could be done in the lab, just by growing bacteria, sequencing the whole genome. But it would be extremely time-consuming, and as I said, we can still only cultivate in the lab a very small fraction of the microbiome. So we developed computational tools building on metagenomic sequencing to get to this resolution. And what we know, first of all, is that, we confirm that we get our first inhabitants from our mothers during delivery and after. Maternal strains are more persistent than environmental ones in our microbiome, suggesting that they are better adapted. And then in a meta-analysis, again, trying to get rid of biases that we also have in the microbiome dataset, so get a bit of a better geographical representation, we see that overall, in the first year of life, the infants are sharing around 50% of their strains with the mother, and it's after, in line with their expansion of their motor activities, that they start to share less with the mother and more with other individuals. And throughout populations, we see that, yes, there are compositional differences, but there are a set of taxa that we tend to get from our mothers, so mostly bifidobacteria and Bacteroides. From an evolutionary perspective, this would make sense. We would say, well, we have co-diversified with our microbiomes, so it makes sense that we are just getting our microbiome through generations. So I said, well, let's actually test that. It has never been shown. So we went on the public media in Belgium, and we recruited 24 families where they had at least 3 generations of women. So the grandma, the mother, and the daughter. Some of them had up to 5. And what we actually saw is that, yes, there were in some cases where we could see the same strain over generations. That's the case in red there. We also had some in gray. That's where it's below our level of detection, so we just don't know whether it was there or not. And then there are also these cases here in blue where there was another strain. So in these cases, this is, again, cross-sectional data. So we don't know whether there was a strain replacement after reproduction, or if it's the grandma that passed the strains directly to the granddaughter. So indeed, we saw some sort of indirect evidence for horizontal transmission of the microbiome, meaning that we said those individuals that cohabitated, so when the grandma lives with the granddaughter or spends more time with her, she just shares more with her. And we think this makes sense, as if we say that infants have a much less diverse microbiome, it's unlikely we're getting everything from our mothers. We must be acquiring strains later on. So that's where we started thinking about the social environment, and that being exposed to other human microbiomes is actually very important. What we saw is that, indeed, we share very little strains with individuals in the same or other populations we've never been in contact with, and we share quite a bit with people in the same household. So talking about horizontal transmission, we see that actually other individuals, so other caregivers, actually play also a role in seeing the infant gut. So we looked at fathers here, which were not sampled in many of the previous studies. And we see that, well, like for mothers, there is this strong initial inoculation, and then strain sharing with the infant decreases. For fathers, that's much more stable. So it starts lower, but it's stable. So at 1 year of age, the infant is sharing the same with the mother and the father. And that's even more important in infants that were born by cesarean delivery, where mothers in our cohort got intrapartum antibiotic prophylaxis, so they had much less of their microbiomes to pass through the kids. So the father in there seemed to take over comparatively. More recently, this was published this year, we looked at also infant-to-infant transmission at the nursery. By improving our tools, I'm not going to get into the details here, we actually can use the same methods that were developed for epidemiology of pathogens, but here for hundreds of species in the microbiome. And just said one example. So we have this Akkermansia mucinifila strain that was transmitted from the mother to the baby Then the baby gets antibiotics, loses the strain, no problem, the mother recedes it. And then at some point, this baby gets the strain actually from the educator in the nursery. But the baby passes it also to another one. And then this baby passes it to the mother and the father at home. So we can actually track, like with longitudinal sampling and our improved methods, we can track transmission of the microbiome, even for species that have never been grown in the lab. Going one step further, we see that so the transmission of the microbiome is associated with host geography, and collaborators show that this also extends to second-degree connections in the social network. So you also share with the friends of your friends. We've been looking a bit also to other microbiomes. That's for the oral one. And there, interestingly, we don't see this strong inoculation from the mother early on. What we see is that it's mostly time-dependent. So the more time you spend with someone, the more you share. And there is more sharing for the oral microbiome than for the gut, which to us makes sense. On the gut, most of the species are anaerobic. They cannot really live for a long time outside our bodies, whereas for the oral, they have saliva as transmission vehicles. And actually, we see that partners share the most. So, well, kissing, surprisingly, plays a role. So just to wrap up my presentation, we started looking at what happens then in disease. So how are these patterns altered? And again, here I'm going to focus on ASD. This was data that we got from the Simons Foundation. Interestingly, they set up this cohort to look at the human genome, but sequencing was so deep that we also got enough microbiome data to analyze, so that was really great for us. And what we saw is that, well, first of all, when we look at differences between children with ASD and neurotypical siblings, is that yes, there are some differences, mostly linked to increased aerotolerant species in the oral microbiome of children with ASD. When discussing this with clinicians, we said that, well, this could be linked to mouth breathing behaviors. And what's something very important here is that this had to be correctly adjusted for diet. There has been lots of discussion in the microbiome field in things with ASD. Some publications actually showed signatures of ASD that were fully explained by diet. So that's something that we were trying to address. But going back to the topic of transmission, what we see is that children with ASD, they just share less with both the father and the mother as compared to their neurotypical siblings. And that's also true, so not only on a binary way, but it's also linked to items in the autism social communication score. So reduced social microbiome transmission is something that we do see in children with ASD. So just finish here, my take-home messages. So the gut microbiome biotransforms a wide range of neuroactive compounds. This could have potential impact on our health, and it's a modulable route of microbiome-gut-brain communication. Something also that we can assess computationally at scale. And when we look at it functionally, we see some more robust associations. And we need a strain level resolution. Also, as in more recent work, we see that our environment and social network also play a role in shaping the composition of our microbiomes. And my last point is that I do think you should include microbiome samples in exposome studies. If you have collected fecal or oral samples, your cohort, I'm very happy to discuss this. And also there, like, consider the factors that are also lengthening the composition of your microbiome. With this, I would like to thank funders, members of my group that are working strongly to make this advance, and our collaborators. So thank you.
At one point, I was thinking of asking you the mapping question again, but I decided everybody would get mad at me. So I have a different question. Related to your case control design, obviously there's the potential for reverse causation because kids who are autistic will have different eating habits than kids who are not, and that could affect their microbiome. Yeah. But within just the cases, you could look at whether or not the microbiome predicts the severity of the phenotype. Have you done any kind of analysis like that?
Actually, we haven't, and that's a good idea. Yes, it's something we could look at. I mean, here, in some sites I would say we don't have enough power, but here we do. So yeah, definitely. I'm going to look at it. Thank you.
I had the same question as Bob, in fact, regarding reverse causality. It's something which is missing sometimes in the field, But I know that you are working on studies that use samples when people are healthy, cognitively, and have prospective data for cognitive trajectories, incidence of Alzheimer's disease, for instance. In your effort, ongoing, I think, to do this very big pooled analysis, it's only case control.
So far, like in the, yes, where we are now, it's case control. That's what we had at the moment. But of course, I mean, having longitudinal data and prospective cohorts would be much better, just that we don't have large data sets yet. So that's also why I'm so interested in including this data, getting the sequencing data, and then looking at that. That's going to let us have a much better picture of what's going on.
Great. And another question, if I have time, is there are a lot of data, and what you are doing is really amazing, but sometimes for people, epidemiologists like me, it's very difficult to have an idea of if we have some convergence of signal or results across disease, or a list of a few species that really seem to be affected, you know, or is it much more complex and it's really signatures which are very specific to a disease? And
yeah, that's a good point. I mean, we are working with data with many features. But to me, the limitation here is that when you look at different studies, those are not consistent. So, and medical doctors are asking us, like, can we have just a range? So we want to know, below what threshold this bacterium is supposed to be good or bad, and so on. And we don't have that yet. I do think that, we just need to have some extra work. Because, I mean, giving some microbiome features that are correlated, and then including it in a broader study, that's something straightforward. But I mean, are we sure, or do we have some real, strong evidence to think that those are something that are meaningful or not? And that's more our limitation at the moment.
Is it okay? I'm Shoji Neki. I'm from National Institute for Environmental Studies in Japan. We're running, children's cohort, and we're planning to do the microbiome for our study. Not 100,000 probably, but at least thousands. But big question is that how stable the microbiomes are, and then also when and what and how many times we have to measure them. So that's a big question for us. And then we do this small interventional study, and then we just put the children under the camp, and then for 30 days, and we measure microbiome before and then after, and just after, and then a month after. Okay. And then microbiome just changes, during that 30 days camp, and then it goes back to the original state after one month. So. Do you have any idea about it?
So I would say, the microbiome can change very quickly. Like the extreme example is antibiotics. You see changes in a day. If we go for drastic dietary changes, we see changes in a week to a month. But all those changes are more superficial. So even if someone went from a vegetarian diet to eating lots of meat, we can very easily identify the sample from that same individual in a longitudinal data set. So there are fluctuations, but there is still strong signature of the individual in there. So I would say that in your study, 1 month and then seeing difference 1 month after, that would make sense. You are going to see differences and you are going to understand patterns, but you can still, the host effect is going to be stronger than what you have in the changes.
Actually, I'm really glad that we talked about the microbiome at this conference, because I think it's really an important part of the exposome. One question is, I seem to recall very early on, people would take a given microbiome and put it into different strains of mice. And then what was constituted is very much strain specific. So is the microbiome really dictated by an individual's genetic background? And the other question is, with the ubiquitous use of antibiotics across the human population, how is that actually interfering or contributing to changes in the microbiome?
Okay. Two great questions. So on the first one, I mean, there are some associations, and in GWAS studies can always some variant. Like for lactose intolerance, that's very clear. For all the rest, I mean, we did also some studies on the twins, UK sets looking at mono versus dizygotic twins. And I mean, host genetics plays some role, but environmental is much stronger than that. On the second one, on antibiotics, I mean, a robust pattern is that in the westernized world, we are having much less microbiome diversity, and it looks like we are losing diversity as compared to non-westernized areas of the world. And yeah, antibiotics plus ultra-processed foods and other variables are probably important on that. But there are no long-term studies, longitudinal studies that are assessing this in detail. So yeah, with more data, we're going to have a much better answer.